Coming to the genetics part 4 session, so we have another topic, that is the pedigree analysis. So scientists have devised an approach called the pedigree analysis to study the inheritance of a certain trait, abnormality or disease. So the pedigree is a chart showing the record of inheritance of a particular trait two or more over ancestral generations of a person, so in the form of a tree. So the pedigree analysis is represented in the form of a tree. So family tree. It is in the form of a family tree. So the pedigree analysis is useful in many ways, like to work out the possible genotypes. To work out the possible genotypes from the knowledge of the respective phenotypes. The second one is it is used to study the inheritance of patterns of a dominant or recessive trait. And the third one is the possible genetic makeup of a person for a particular trait is also known with the help of this pedigree analysis. So here some symbols are standard symbols are there that is used in our pedigree analysis. So the empty circle represents a male. The empty circle represents a male person. And the empty square represents a male person, and the empty square represents a female person. So the empty square represents a male person, and the empty circle represents a female person. An empty diamond shaped represents the sex unspecified. The sex unspecified. And a line between a square and a circle, it represents a mating. It represents mating. And here a two horizontal line, so between the square and the circle represents mating between relatives. Mating between relatives. And so these empty square and circle, so these are the parents. So these are the children, the female children and the male children. And here these are the parents with the shaded square. So the parents with male child affected with the disease. The parents with male child affected with disease. So here the shaded square, the shaded circle, and the shaded this one represents the affected individuals. The empty square represents the person, and the shaded circle represents represents the affected individuals. And here it is a 5. So this 5 represents the 5 unaffected offsprings. The 5 unaffected offsprings. So these are some symbols that are used in pedigree analysis. And now the next topic is genetic disorders. So genetic disorder. So it is a genetic disorder. So the genetic disorder is a syndrome or disease. It is caused by the abnormality in individual's DNA. So genetic disorders. So these genetic disorders are, it is a syndrome or disease caused by the abnormality in individual's DNA. Abnormality. So in individual's DNA. Abnormality in individual's DNA. So a number of disorders have been found associated in human beings. So a number of diseases in human beings have been found to be associated with the inheritance of changed or altered genes or chromosomes. So these genetic disorders are broadly classified into two categories. So the genetic disorders are classified into two categories. So one is the Mendelian disorder. Mendelian disorders. And the second one is the chromosomal disorders. So chromosomal disorders. So coming to the Mendelian disorders. So Mendelian disorders. So these Mendelian disorders exhibit the Mendelian type of inheritance. Mendelian disorders exhibit Mendelian type of inheritance. Mendelian disorders. So exhibit Mendelian type of inheritance. So it is caused by the mutation, single mutation in DNA. So also called the monogenic diseases. So also called monogenic diseases. Monogenic disease. So it may be autosomal. I mean these disorders may be autosomal or sex linked. So may be autosomal or sex linked. Autosomal or sex linked. So examples for these Mendelian disorders are examples for the Mendelian disorders are hemophilia, sickle cell anemia, phenylketonuria, thalassemia, so color blindness, and last one is cystic fibrosis. Fibrosis. So these are the disorders that are under the Mendelian disorders. So first of all, we know about we discussed about the hemophilia. So in the previous session we discussed about the hemophilia. So it is also called bleeder's disease and also called the Royal's disease, right? So it is of two types, hemophilia A and B. So most of the 80% of the people represents the hemophilia A cases. So hemophilia A, so it is due to the lack of functional clotting factor 8 and it is due to the lack of functional clotting factor 9. So when a person with hemophilia is injured, the bleeding is prolonged, so the clot is slow to form. So that's why it is called the hemophilia, bleeder's disease, and it is also called the Royal's disease. So coming to the second one is the sickle cell anemia. Sickle cell anemia. So sickle cell anemia, it is an autosomal genetic disorder. So due to the affected gene on the chromosome 11. So affected gene on chromosome 11. So it appeared in homozygous condition. Exhibits in homozygous condition. Homozygous conditions. So the symptoms for sickle cell anemia is the physical weakness. The symptoms are the physical weakness, paired organ damage, so due to the internal bleeding, and even paralysis also. So the symptoms are physical weakness, paired organ damage, due to the internal bleeding, so and even paralysis also. So it is caused by the replacement of the glutamic acid. So it is caused by the replacement of glutamic acid by valine. So these glutamic acid and the valine are the amino acids. So in the amino acid composition, so there is a replacement of the glutamic acid by valine, causing the sickle cell anemia. So due to the replacement of the glutamic acid by valine, so it causes the RBC become sickle shaped. So the RBC become sickle shaped. The RBC becomes sickle shaped. So actually the structure of RBC is like that. So this, due to the replacement of the glutamic acid by valine, so this RBC becomes sickle shaped and rigid. Sickle shaped and rigid. Sickle shaped and rigid. So the phenotypes for sickle cell anemia for homozygous individuals, so it is these are controlled by so the sickle cell anemia is controlled by a single allele called HBS, HBA genes. So controlled by HBS and HBA. And these phenotypes for sickle cell anemia for homozygous individuals are, so phenotype. So for homozygous individuals, are HBS and HBS. And for heterozygous individuals, so individuals, so HBA and HBS. So but these heterozygous individuals appear unaffected. The heterozygous individuals appear unaffected, but still they are carriers of this. But heterozygous individuals are still carriers of this disease, and also the heterozygous individuals are more resistant to the malaria. So these individuals are more resistant to malaria. So this is about the sickle cell anemia. So and the second one is third one is the phenylketonuria. So it was discovered by A. Fowling. So phenylketonuria was discovered by A. Fowling. Discovered by A. Fowling. So it is an autosomal genetic disorder. Phenylketonuria, it is an autosomal genetic disorder due to the defective gene on the 12th chromosome. So due to defective gene on 12th chromosome. 12th chromosome. So this phenylketonuria is caused by the enzyme called phenylalanine hydroxylase. So phenylalanine hydroxylase. So this due to the mutations, this phenylalanine hydroxylase cannot produce. So this phenylketonuria is caused by the phenylalanine hydroxylase. Phenylalanine hydroxylase. So due to the mutations, this enzyme cannot be produced. Due to the mutation, this enzyme cannot be produced. So whenever this enzyme cannot be produced, this phenylalanine hydroxylase cannot convert it into the tyrosine. So it is cannot converted into tyrosine. So this tyrosine is converted into phenylpyruvic acid. Phenylpyruvic acid. So whenever the tyrosine is converted into phenylpyruvic acid, so this pyruvic acid is accumulated in our body fluids. this phenylpyruvic acid is accumulated in our body fluids. So whenever this phenylpyruvic acid is accumulated in our body fluids, it leads to some disorders. Some disorders are mental retardation, reduction in hair, reduction in skin pigmentation. Reduction in skin pigmentation. So these are some disorders whenever the phenylpyruvic acid is accumulated in our body fluids. So one is the mental retardation, reduction in hair, and reduction in our skin pigmentation. So this is about the phenylketonuria. So next one is the thalassemia. So it is an autosomal genetic blood disorder. So caused by the excessive destruction or degradation of the RBC. So caused by the excessive destruction or degradation of RBC. So due to the abnormality in the Hb molecule. So normally the Hb molecule. So the Hb molecule is composed of 4 polypeptide chains. The Hb molecule is composed of 4 polypeptide chains. 4 polypeptide chains. So 2 alpha chains and 2 beta chains. So these 2 alpha and 2 beta are arranged into a heterotetramer. So normally the Hb molecule is composed of 4 polypeptide chains, 2 alpha and 2 beta globin chains arranged into a heterotetramer. So but in case of thalassemia patients, so this Hb molecule, there is a defect. So there is a defect in either 2 alpha or 2 beta chains. There is a defect in alpha or beta chains causing production of abnormal Hb molecules. So defect in these 2 chains causing abnormal Hb molecules. So resulting what? Anemia. So once again I repeat this. So normally the Hb molecule is composed of 4 polypeptide chains. So 2 alpha and 2 beta globin chains. These 4 are arranged into a heterotetramer. So but in case of thalassemia patient, so there is a defect in in either alpha chain or beta chain. So causing the production of abnormal Hb molecules resulting anemia. So this anemia condition, it is the characteristic feature of this thalassemia patient. Right? So the thalassemia is classified into 2 categories. So 2 types. So thalassemia, alpha thalassemia, and second one is the beta thalassemia. Alpha thalassemia and beta thalassemia. So the alpha thalassemia is controlled by HbA1 and HbA2 genes. HbA1 and HbA2. And beta thalassemia. So it is also called Cooley's anemia. Cooley's anemia. Cooley's anemia. So it is controlled by a single gene, HBB. So HBB. So on chromosome 11. So it is on chromosome 16. It is on chromosome 16 and HBB gene on the chromosome 11. So this beta thalassemia, the most common type of thalassemia is the beta thalassemia. The most common type of the thalassemia is the beta thalassemia. So this is about the thalassemia. So alpha thalassemia and beta thalassemia. And now coming to the next one is the color blindness. Color blindness. So in the previous class we discussed about this color blindness. So it is a recessive sex-linked disorder, right? It is a recessive sex-linked disorder. So what is the character of this color blindness? So inability to distinguish between the red and green color. So inability to distinguish between red and green color. Inability to distinguish between the red and green color. So the red color blindness. The red color blindness is called protanopia. So this is a red color blindness. Next one is the deuteranopia. So it is a green color blindness. Green color blindness. So one more disease is also there, tritanopia. Tritanopia. So it is a blue color blindness. So the color blindness is of three types. So one is the protanopia, second one is the deuteranopia, third one is the tritanopia. So protanopia. So in this protanopia, it is a red color blindness. Here the persons cannot identify the red color. Cannot identify the red color. So in case of deuteranopia, the person cannot identify the green color. So these protanopia and deuteranopia, these are the sex-linked recessive disorders. So apart from the tritanopia. So here the tritanopia, it is an autosomal disorder. So it is an autosomal disorder. It is an exemption. So it is an autosomal disorder. So these two are the sex-linked recessive disorder. So how can we identify the color blindness? Here's one test is there. Ishihara color blind test. Ishihara color blind test so is conducted to diagnose the disease. Ishihara color test to diagnose the disease. To diagnose the disease. So it was the color blindness is already discussed in our previous class. That was I skipped in this session. So it is the brief introduction about the color blindness. It is of three types: protanopia, deuteranopia, and tritanopia. So protanopia, it is a red color blindness. Deuteranopia, it is a green color blindness. Tritanopia is a blue color blindness, but it is an autosomal disorder. The Ishihara color blind test is conducted to diagnose the disease. So coming to the next topic, next disease in our Mendelian disorders are cystic fibrosis. Cystic fibrosis. It is an autosomal recessive disorder. So first of all, it is an autosomal recessive disorder. So there is the affected gene is located on the affected gene is located on short so fine. So that influences sodium and chloride and sweat. So the symptoms are increased sodium and chloride in sweat. And the second one is mucus become viscous and sticky. And the mucus clogs in lungs, pancreas and alimentary canal. So etc. These are the symptoms. Increased sodium and chloride in sweat. So the mucus become viscous and sticky, and the mucus clogs in lungs, pancreas and alimentary canals, etc. So this is about the cystic fibrosis. So these are the Mendelian disorders. So one is the hemophilia. So second one is the phenylketonuria, sickle cell anemia, color blindness, thalassemia and the cystic fibrosis. So coming to the chromosomal disorder. So these chromosomal disorders are caused by the addition or deletion of chromosomes or by the accidental exchange of the chromosomal bits during meiosis. So caused by the addition, deletion or accidental exchange of chromosomal bits during meiosis or non-disjunction. So the chromosomal disorders are caused by the addition or deletion or accidental exchange of chromosomal bits during meiosis or non-disjunction. And so when a chromosome is deleted from a pair, so that is represented by monosomy. So 2n minus 1. So when a chromosome is added to a pair, so the condition is called a trisomy. So represented by 2n plus 1. So when a chromosome is deleted from a pair, that is called 2n. That is represented by 2n minus 1. When a chromosome is added to a pair, that is called condition is called the trisomy. That was represented by 2n plus 1. So these chromosomal disorders are again two types: autosomal disorders Allosomal disorders. Autosomal disorders. So, the chromosomal disorders are classified into two types, allosomal disorders or autosomal disorders. So, first of all, we discuss about the allosomal disorders. So, these are of two types. So, one is the Klinefelter syndrome. Second one is the Turner syndrome. Turner syndrome. So first of all, we discuss about what is Klinefelter syndrome. So the Klinefelter syndrome, it is a genetic condition caused by the trisomy. So caused by trisomy 23rd pair. Trisomy 23rd pair. So karyotype of Klinefelter syndrome is karyotype is so 47 XX5. 47. I mean the humans have 46 chromosomes, right? So here in the Klinefelter case, an extra X chromosome is present along with the XY chromosome. So in case of Klinefelter's males, especially males, in case of Klinefelter's males, an extra X chromosome is present along with the XY chromosome. So it is caused by the trisomy 23rd pair. So an extra X chromosome is present along with the XY chromosome. So the symptoms are so hypogonadism, reduced fertility, and the gynecomastia. So what is gynecomastia? So slight enlargement of the breast in females, that condition is called gynecomastia. So these are the symptoms: hypogonadism, reduced fertility, and the gynecomastia. So after that, the Turner syndrome. So Turner syndrome, it is due to the monosomy. Caused by the monosomy. Monosomy 23rd pair. So karyotype is so 45X. So here one X chromosome is lost. One X chromosome is lost. Here one X chromosome is lost. The symptoms are short stature, so small round head. That's it. So these are the symptoms that are present in the Turner's syndrome. So the Klinefelter syndrome and the Turner syndrome, these are under the allosomal disorders. So now the next one is the autosomal disorders. So these autosomal disorders, so first of all, Down syndrome. So before going to know about the autosomal disorders, so these are common in children. So these disorders are common in children born to the women, so who receives their babies rather later in their reproductive phase. So the autosomal disorders are common in children, so born to women who receive their babies late in their reproductive phase. So these disorders are of so first one is the Down syndrome. Second one is the Edwards syndrome. Third one is the Potter syndrome. Third one is the Cradle-Short syndrome. And fifth one is the chronic myelogenous leukemia. Chronic myelogenous leukemia. So these are the autosomal disorders. So first of all, we discuss about what is the Down syndrome. What is Down syndrome? So it is a genetic condition. So it is a genetic condition caused due to the trisomy 21st pair. So caused due to the trisomy 21st pair. 21st pair. So karyotype is 47 XX plus 21. Karyotype is 47 XX plus 21. So the Down syndrome. The symptoms are cardiac abnormalities, kidney malfunctions. So these are the two symptoms in the Down syndrome: cardiac abnormalities and kidney malfunctions. And the affected individuals, the child dies. So before going to the in infant stage or early years of life, the child dies in childhood. In infant stage or early years of life. So this is the Down syndrome caused due to the trisomy 21st pair. Karyotype is 47 XX plus 21. The symptoms are the cardiac abnormalities and the kidney malfunction, and the child dies in an infant stage or early years of life. So second one is the Edwards syndrome. So it is caused due to the trisomy 18th pair. Caused due to the trisomy 18th pair, and the karyotype is 47 XX plus 18. 47 XX plus 18, and the symptoms are heart and kidney defects, brain and spinal abnormalities, poorly developed eyes. So these are the symptoms: heart and kidney defects, brain and spinal abnormalities, and the poorly developed eyes. So the newborn baby dies within weeks after birth. The newborn baby dies within weeks after birth. So Edwards syndrome, it is caused due to the trisomy 18th pair. So karyotype is 47 XX plus 18. So the symptoms are heart and kidney defects, brain and spinal abnormalities, and the poorly developed eyes. And now the third one is the Potter syndrome. So this Potter syndrome is caused due to the trisomy 13th pair. trisomy 13th pair. So it is caused due to the chromosomal abnormalities. So it is due to the chromosomal abnormalities. Due to the delay in the intellectual or physical development. So delay in physical. So karyotype is 47 XX plus 13. So delay in physical and intellectual development. So the symptoms are heart and kidney defects. Cardiac abnormalities. So these are some disorders in the Pato syndrome. So it is caused due to the 13th pair. So karyotype is 47XX plus 13. And now the next one is the Cry-Do-Chart syndrome. Cry-Do-Chart syndrome. So it is caused due to the partial deletion of the chromosome 5. Due to the partial deletion of the chromosome 5, so also called 5p monosomy. 5p monosomy. So the symptoms are short stature, small head, unusual facial features, etc. So the child dies in infancy or childhood. The affected child dies in infancy or childhood. So the cry-do-chart, one more symptom is the cat-like cry. Short stature, cat-like cry, small head, unusual facial features. These are the symptoms that are present in the cry-do-chart syndrome. So coming to the last but not the least, chronic myelogenous leukemia. CML, chronic myelogenous leukemia. In certain types of cancer like CML, so it is also called chronic granulocytic leukemia. So also called chronic granulocytic leukemia. So in this leukemia, it is caused by the breaking chromosome 9 and the chromosome 22. So these chromosome 9 and chromosome 22 break off and switch places with each other. Exchange. So their places. So here by translocation. So breaking of chromosome 9 and 22 with each other. So for forming. So whenever these two chromosomes break up and switches or exchange their places, forming a short 22 chromosome and long arm of 9th chromosome. Long arm of 9th chromosome. So the short 22 chromosome is called a Philadelphia chromosome. The short, the 22 chromosome is called a Philadelphia chromosome. Philadelphia chromosome by translocation. So, and the chromosome is called the Philadelphia translocation, and the translocation is called the Philadelphia translocation. How it is formed? By the translocation of the 9th and 22 chromosome. So, they switches places with each other. So, that's why they are formed. So, Philadelphia translocation. Philadelphia translocation. Philadelphia translocation. So this Philadelphia chromosome produce an enzyme called tyrosine kinase. So produce an enzyme called tyrosine kinase. So this tyrosine kinase produce uncontrolled cell cycle progression. So this tyrosine kinase causes uncontrolled cell cycle progression. Uncontrolled cell cycle progression, so leading to cancer. Leading to cancer. So this tyrosine kinase causes the uncontrolled cell cycle progression, so leads to cancer or CML. So the CML, it is a chronic myelogenous leukemia, so also called the chronic granulocytic leukemia. So the chromosome 9 and 22 switches are break off and switch places or exchange their places by translocation. So the short arm chromosome is called the 22 chromosome and the long arm chromosome is called the 9th chromosome. So the 22 chromosome is called the Philadelphia chromosome. So it is formed by the translocation. So that's why it is called Philadelphia translocation. So this Philadelphia chromosome produce an enzyme called the tyrosine kinase. So this tyrosine kinase causes the production of an enzyme called amina, causes the production of uncontrolled cell cycle progression. So this leads to the cancer. So this is about the chronic myelogenous leukemia. So these are the disorders that are under the chromosomal disorders. So chromosomal disorders are the two types: autosomal disorders and allosomal disorders. Autosomal disorders are the Klinefelter syndrome and the Turner syndrome, and allosomal disorders are the first one is the Down syndrome, second one is the Edwards syndrome, third one is Porter's syndrome, Krabbe-Chaud syndrome, and last one is the chronic myelogenous leukemia, that is represented as in a short form, that is the CML. So this is about the genetic disorders. This is about the genetic disorders. So in our next session, so we have in our genetics chapter one more two topics are there. So one is the Human Genome Project and last one is the genetic DNA fingerprinting. The Human Genome Project and the DNA fingerprinting. So these two topics are discussed in our next session, that is part 5. So thank you very much.
Good morning, students. Myself Uma, KZBB Banganapalli. I'm working as a PGT in chemistry. In this session, we are discussing the lesson continuation biomolecules. In that, two topics we are discussing today. So the first topic is proteins. So the word proteins. The word protein is derived from Greek word that is proteos. The word protein is derived from Greek word that is proteos, which means primary or prime importance. Primary or prime importance. All the prime polymers are alpha amino acids. All proteins are polymers of alpha amino acids. Proteins are the most abundant biomolecules of the living system. Proteins are the most abundant biomolecules of the living system. Okay, what are the sources of the proteins are milk, cheese, peanuts, fish, meat, etc. These are the sources of the proteins. They are present in every part of the body and also required for growth and maintenance of the body. Okay, this is the introduction of the proteins. Again, I repeat it. The word protein is derived from Greek word that is proteos. Proteos means primary or prime importance. All proteins are polymers of alpha amino acids. Proteins are polymers of alpha amino acids. Proteins are most abundant molecules of the living system. The sources of the proteins are milk, cheese, peanuts, fish, meat, etc. Okay, they are present in every part of the body and also required for growth and maintenance of the body also. Okay, next we are discussing the structure of proteins. What is the structure of proteins? How many types of structures present in proteins? In that we discussed what is peptide bond or peptide linkage. The peptide bond or peptide linkage formed between one of the COOH group, that is carboxylic group, and NH2 group, that is amine group. Carboxylic group, one amine group combines to give one peptide linkage by the elimination of water molecule. Such type of bond is known as peptide bond. Okay, so let us see how to form a peptide bond. See here, two amino acids are there. One is NH2CH2COOH, that is one of the amino acid, and second also amino acids. The amino acids of the protein first one is glycine and second one is valine. First one is glycine and second one is valine. These two amino acids combine by the loss of water molecule to form a peptide linkage. See here, first molecule, amine group and carboxylic acid group, both groups are present. At the same time, second molecule also amine group and carboxylic acid group both present. But by the reaction, these one of the carboxylic acid and another amine group combines and loses water molecule to form peptide. In COOH group, hydrogen is replaced, and at the same time NH amine group also they loses hydrogen. Totally OH present in COOH, NH2 present in H. Both are removal of water molecule to form one amide group, that is CONH group is formed, that is amide group, that also called as peptide linkage. So it is first molecule glycine, second molecule is alanine, combines to give final compound glycyl alanine. Short formly we are write GLYALA. Okay, amino acids are we are shortly named to 3-letter symbol. So these are 3-letter symbols of the amino acids. So here in this equation amide group is formed. That amide group is known as peptide linkage. Here dipeptide, tripeptide, pentapeptide, hexapeptide, and so on. Polypeptides are so formed. Dipeptide. How to form dipeptide? When a carboxylic group of glycine combines with the amino group of alanine, you get dipeptide of glycyl alanine formed. Okay, this is dipeptide. In this equation we are seeing the dipeptide compound. Okay, tripeptide means in any of the 3 amino acids combines to form tripeptide. Hexapeptide, pentapeptide, polypeptides are formed like this. Okay, next, what is the structure of the proteins? Let us discuss what is the structure of proteins. Okay, proteins are polymers of alpha amino acid. We are already know proteins are polymers of alpha amino acids. A polypeptide chain with more than 100 amino acids. A polypeptide chain with more than 100 amino acids having a molecular mass or molecular weight higher than the 10,000 units. Higher than the 10,000 units is called protein. Again I repeated. Proteins are polymers of alpha amino acids. A polypeptide chain with more than 100 amino acids having molecular mass higher than 10,000 units is called protein. Okay, what are the examples of the protein? Egg yolk, blood serum, hemoglobin, myoglobin are the examples of the proteins. Okay, next, classification of proteins. Proteins are classified into 2 types. They are fibrous proteins and globular proteins. So proteins are classified into 2 types. One is fibrous protein and second one is globular proteins. On these two are based on the molecular shapes are classified. So first one, let us discuss first one, fibrous protein. What is the fibrous protein? These proteins contains linear thread-like polypeptide chains which are arranged by side to form fibers. Okay, again I have repeated. These proteins contains linear thread-like polypeptide chains which are arranged side by side to form fibers. Okay. Next point. In these proteins, various polypeptides are held together. In these proteins, various polypeptides are held together by hydrogen bond. It is a very important point to saying like in these proteins, various polypeptide bonds are held together by hydrogen bond. Okay, so and also disulfide bonds also present in fibrous protein. So these fibrous proteins are generally insoluble in water. These fibrous proteins are insoluble in water. The examples of the fibrous proteins are keratin and myosin. Okay, keratin and myosin are the examples of the fibrous protein. This is about the fibrous proteins. Okay, next go to second classification, that is globular proteins. Globular proteins means the protein in which polypeptide chains are very tightly folded into compact spherical form are called globular protein. Okay, again repeated, the proteins in which polypeptide chains are tightly folded into compact spherical form are called globular proteins. Next point, intramolecular forces are, so in between globular protein structure, intramolecular forces are presented. Okay, so these means globular proteins are water soluble. So fibrous proteins are water insoluble, globular proteins are water soluble. Example for globular proteins are insulin and albumins. Insulin and albumins are the examples for globular proteins. Okay, this is the classification of the proteins. Okay, next go to structure of the protein. What is the structure of the protein? How they studied in the biomolecules. The structure and shape of the protein. Structure and shape of the protein is studied under 4 different levels. Structure and shape of the protein is studied in 4 different levels. First one, in that first level is primary structure. So primary structure, proteins may be one or more peptide chains contains. Proteins may have one or more polypeptide chains are present. Okay. Second point, it gives sequence of amino acids. It gives a sequence of amino acids in given polypeptide chain and location of disulfide linkage if present. That means each amino acid in polypeptide chain and location of bisulfate linkage if present. Okay, third point, any changes in primary structure. Any change in the primary structure results in the formation of different protein. If any changes present in the primary structure, different proteins are the available. Okay, this is the primary level of the first structure of proteins. Okay, primary structure. First level is primary structure. Proteins may have one or more polypeptide chains. Okay, next, it gives a sequence of the amino acids present in polypeptide chain. At the same time, this disulfide linkage also. Okay, if any change in primary structure results the formation of different proteins. This is the first primary structure. Second level of the primary structure of proteins, that is secondary structure. It refers shape of long polypeptide chain. It refers shape of long polypeptide chain. Okay, next, these are found to exist in 2 different types of structures. So in secondary structure, 2 types of structures are possible. That is, first one is alpha helix. Alpha helix is the first type of structure, and second one is beta pleated sheet structure. In these structures, arises due to regular folding and regular folding of backbone of the polypeptides. These 2 structures, how to form due to regular folding of backbone of the polypeptide chains. Okay. Next, let us discuss structures of alpha helix and beta pleated sheet structure. In alpha helix structure, the polypeptide chain forms all possible hydrogen bonds. In alpha helix, the polypeptide chain forms all possible hydrogen bonds. How to form? By twisting into a right handle helix. Okay, this is the structure of helix. In alpha helix, hydrogen bonds are formed between NH group of amino acid in one turn with carboxylic group of the adjacent turn of the helix in the same chain. Again I repeat it. In alpha helix, hydrogen bonds are formed between NH group of amino acid in one turn with the carboxylic group of adjacent turn of the helix in the same chain. That means one of the amine group and one of the carboxylic group will be twisted. Then we get alpha helical structure. Okay, again I repeat it. Alpha helix means a polypeptide chain. All possible hydrogen bonds are twisting into a right handed. So it is alpha helix. In alpha helix, hydrogen bonds are formed between one of the hydrogen group of amine group of amino acid in one turn with carboxylic group of adjacent turn on the helix in the same chain. Okay, this is alpha helix. And go to second one, beta pleated sheet structure. So all peptide chains are completely stretched and put together side by side. Again I repeated, in beta pleated structure, all peptide chains are completely stretched and put together side by side. These are held together by intramolecular hydrogen bond. In this beta pleated structure, held by the intramolecular hydrogen bonds. Okay, this is about the beta pleated structure. Again I repeated, second level of the protein or structure of proteins, that is secondary structure. It refers to the shape of the long polypeptide chain. These are found to exist in two different types of structures. Two different structures will be formed in secondary structures, that is alpha helix and beta pleated sheet structure. So these structures arise due to regular folding of backbone of polypeptide chain. So let us discuss one by one. Alpha helix structure. The polypeptide chain forms all possible hydrogen bonds by twisting into a right-handed helix. Okay, so hydrogen bonds are formed between amine group of the amino acid in one turn with the carboxylic group of the adjacent turn on the helix in the same chain. So this is the alpha helix. And in beta pleated structure, in beta pleated structure, all peptide chains are completely stretched and put together side by side. These are held together by the intramolecular hydrogen bond. As beta structure resemble the pleated folds of drapery, it is known as a beta pleated sheet structure. See here the structure of alpha helical of protein like this. In between this structure, hydrogen bonds are present. So at the same, these hydrogen bonds are twisting in right-handed helix. That here hydrogen bonds are formed between NH group of amino acid and carboxylic group of adjacent turn of the helix. This is the alpha helical structure of proteins. Okay. Next, second, beta pleated sheet structure. Next, beta pleated sheet structure of proteins. In this beta pleated sheet structure, proteins are held together. That means peptide link chains are completely stretched and put together by side. Okay, in between these peptide chains, intramolecular hydrogen bonds are present. See, hydrogen. One of the amine group and one of the carboxylic acid formed between like hydrogen bonds. These total peptide linkages are stretched and put together by side. This is the structure of the beta pleated structure protein. Okay. Next, go to third level of the structure of protein, that is tertiary structure. It presents three-dimensional folding and bonding of secondary structure, resulting in the formation of fibrous and globular proteins. Again I repeated, it represents three-dimensional folding and bonding of secondary structure, resulting in the formation of fibrous and globular proteins. The various linkages responsible for the tertiary structure. In tertiary structure, there is the responsible for linkages, that is one hydrogen bond present in this tertiary structure, and second one is the disulfide linkage also formed in the tertiary structure. At the same time, van der Waals forces and electrostatic force of attraction is also present in tertiary structure. Again I repeated, the various linkages responsible for tertiary structures, that is hydrogen bond formed and disulfide bonds also formed. At the same time, van der Waals and electrostatic force of attraction is also formed between the tertiary structure. This is the tertiary structure. And quaternary structure. The proteins may have more than one polypeptide chains called subunits. So in tertiary structure, some of the proteins are composed to or more polypeptide chains is called subunits. Okay, the spatial arrangement of these subunits with respect to each other is known as quaternary structure. So this is about quaternary structure. So let us see quaternary structure. The proteins may have more than one polypeptide chains is called subunits. The spatial arrangement of these subunits with respect to each other is known as quaternary structure. This is total about the proteins and classification of proteins and structures of the proteins. And what are the structures? The proteins. See here, this is the structures of the proteins. First, primary structure, and second one secondary structure, and third one is tertiary structure, and fourth one is quaternary structures. Structures are present like this. Okay, let's next go to denaturation of proteins. Denaturation of proteins. So denaturation of proteins means the process that brings about changes in physical as well as biological properties of the proteins is called denaturation of the proteins. Again I repeated, the process that brings about changes in physical and biological properties of the proteins is called denaturation of the proteins. Okay, so this is the denaturation of the proteins. Next, chemically, denaturation does not change primary structure but changes in the secondary and tertiary structures of the proteins. So the chemically, denaturation does not change because primary structure but changes the secondary structure and tertiary structures of the protein. Okay, so explain the denaturation of the protein in step by step. First one is denature of proteins can be caused by changes in pH, temperature, or by adding another denaturing agent. Denaturation can be caused by changes in the pH, temperature, or by adding any denaturing agent. Okay, next, denatured protein loses its biological activity. Denatured protein loses its biological activity. Okay, next, during denaturation, the protein molecule uncoils into a more random conformation leading to precipitation. That means here biological activity will be loses in denatured protein. How to deactivated means a more random conformation leading to precipitation. That is the reason for denatured protein loses their biological activity. Next, denaturation may be reversible or irreversible. So denaturation may be reversible or irreversible. So examples of the irreversible denaturations are the coagulation of egg white on boiling. The coagulation of egg white on boiling. So second one, curdling of milk. Curdling of milk, these two examples are the irreversible denaturation. Again, next point, renaturation. Renaturation is reverse of denaturation. That means all are above we are discussing denaturation process, reverse of the renaturation process. Okay, so total this about the denaturation of the proteins. This is very important for 4 marks, at the same time final exams also. Again I repeated, denaturation of the proteins, the process that brings about changes in physical as well as biological properties of the proteins is called denaturation of proteins. Chemically, denaturation does not change the primary structure but changes the secondary and tertiary structures of the protein. Okay, next, denaturation of protein can caused by changes in pH, temperature or by adding any other renaturating agent. Okay, next, denatured protein loses its biological activity. During denaturation, the protein in molecule uncoils into a more random conformation leading to precipitation. Okay, next, denaturation may be reversible or irreversible. Examples of the irreversible denaturation: coagulation of egg white on boiling and curdling of milk. These two examples. Renaturation is the reverse of the denaturation. Okay. Next topic is amino acids. This is total about the proteins. Next we are going to amino acids. But what is the meaning of the amino acids? So carbon compound contains both NH2 group, that is amine group, carboxylic group, that is COOH group, are present in one molecule is called amino acids. Amino acids means carbon compounds containing both NH2 and COOH group are called amino acids. These are building blocks of the proteins. Okay, these are the building blocks of the proteins. Example, glycine, alanine. So many amino acids are there in that examples, glycine and alanine. So totally in naturally occurring amino acids are nearly 21 amino acids are naturally occurring. In that here we are seeing so many natural amino acids in that. So some amino acids are right here. Here the general molecular formula of amino acids are COOH, CH, NH2. COOH and CH2, NH2 is the general formula of the amino acids. In CH2, one of the hydrogen are replaced by any alkyl group: hydrogen, ethyl, methyl, propyl, butyl, and so on. So number of alkyl groups increased, the name of the amino acid will be changed. Okay, let us see first one. Glycine, name of the acid glycine, and characteristic feature side chain. That means CH2 group, one of the hydrogen is added to the chain, that is glycine. So 3-letter symbol is GLY. 3-letter symbol is GLY. Short form we are coded G. Okay, capital G. So first amino acid is glycine. In alkyl group, hydrogen is present. 3-letter symbol is GLY. G is the code of the letter. Next, second amino acid, that is alanine. So alanine is the second amino acid in alkyl group. Alkyl group means R, CH3 group is replaced. Okay, so it is shortly give the symbol ALA. The code is A. So next, third one, valine. Valine is one of the essential amino acid. Okay, so valine is the one of the essential amino acids. In this structure, alkyl group, that means CH3 taken twice, CH is the branch. So it is shortly given to VAL and the code is V. So next, leucine. Leucine is the amino acid. In this structure, we are one of the carbon atoms. That means CH2 group is added to valine group, valine amino acid. So this is leucine. Shortly we are giving to LEU. So coded is L. Okay. Next, isoleucine. Isoleucine is the fifth amino acid. It is also very essential for us. So in iso means we know that is CH3CH, CH3CH. So this is isoform. Isoleucine, short form of this 3-letter symbol of the isoleucine is ILE. In code letter is I. Next amino acid, arginine. Arginine means the side chain structure like this, and the 3-letter symbol is ARG, and the coded letter is R. Okay, next amino acid, lysine. It is also a very important amino acid or essential amino acid. So it is given by the star letter. star symbol. So here NH2, CH2 taken 4 times are the branch present in amino acid. This 3 letters symbol is Y, LYS. LYS is the 3 letter symbol and the code is K. Okay, this is the 7th amino acid, lysine, essential amino acid also. Okay, next 8th glutamic acid. Glutamic acid, it is an acid which is present in, that is CH2, COOH, like a chain, side chain in amino acid. So it is 3 letter symbol is GLU. It is coded E. Letter is E. Next amino acid, aspartic acid. Aspartic acid is the next amino acid. In this aspartic acid, amino acid, CH2COOH group is present as a side chain or alkyl group. This 3 letter symbol is ASP. Coded letter is D. Okay. So next amino acid, glutamine. Glutamine is the next amino acid. In this amino acid, the structure of alkyl group NH2COCH2CH2 is the alkyl group present in amino acid. The 3 letters symbol of the glutamine is GLN, and the short code of the glutamine is Q. Okay, next amino acid, asparagine. Asparagine is the next amino acid. The structure present in the amino acid by the replace of alkyl group, that is NH2CONH2. Asparagine is the amino acid. This 3 letter symbol is ASN. ASN is the 3 letter symbol and the code letter is N. Asparagine is code letter is N. Next amino acid, threonine. Threonine amino acid, it is also very essential for ours. Okay, next the structure of threonine by alkyl group. Here alkyl group is CH3CHOH, and 3 letters amino acid symbol is THR. THR is a 3 letter symbol and the code letter is T. Threonine T code letter. Next serine. Serine is also one of the amino acid. In this amino acid, alkyl group OHCH2. OHCH2 is the alkyl group. The 3 letter symbol is SER. SER the 3 letter symbol and the code letter is S. Serine. Serine SER 3 letter symbol. S is the code letter. Next amino acid, cysteine. Cysteine is the amino acid. In this amino acid, alkyl group is SHCH2. SHCH2 the alkyl group present in cysteine. 3 letter symbol is CYS. CYS is the 3 letter symbol and short code of the letter S. These short code letters represented by proteins in short form. Okay, next methionine. Methionine also one of the essential amino acid. In methionine, the alkyl group is CH3S, CH3, CH2 and the sulfide group is present in threonine. So the 3 letter symbol of the threonine MET is the 3 letter symbol of the threonine. So the code letter is M. Okay, next amino acid, phenylalanine. Phenylalanine is also essential amino acid. The alkyl group in phenylalanine is C6H5CH2, means phenyl group is benzyl group is present in phenylalanine. The 3 letter symbol of the phenylalanine is PHE and short form of the short code of the phenylalanine is F. Okay, next tyrosine. Tyrosine is the next amino acid. In this amino acid, alkyl group is OHC6H4CH2. OH, that is OH group present in para position. OH group present in para position, C6H5CH2. The 3 letter symbol is TYR. TY are the 3-letter symbol and the code letter is Y. Okay. Next, tryptophan. Tryptophan is the next amino acid. It is also an essential amino acid. So this is the structure of the alkyl group present in amino acid, this one, and the short 3-letter symbol is TRP and the code letter is W. Okay. Next, histidine. Histidine is the essential amino acid and the alkyl group structure is like this, and the short form of 3 letters form of the amino acid is HIS and the letter code is H. Okay. Next, proline. Proline, it is also amino acid. In this proline, alkyl group structure like this. So the 3 letters short form of the amino acid is PRO and code letter is P. So these are all amino acids. Okay. In this amino acids, totally 10 amino acids we are given, very important and also essential amino acids. They are valine, also amino acid, essential amino acid, leucine, also essential amino acid, isoleucine, arginine. Next, threonine, methionine, phenylalanine, tryptophan, histidine. So these are all essential amino acid given in table like stars. Okay. So this total about the types of naturally amino acids present in table. Okay. So this is the table. Next, classification of amino acids. Next, classification of amino acids. The amino acids are classified into 2 types. They are essential amino acids and non-essential amino acids. Okay, so amino acids are classified into 2 types. One is essential amino acid and second one is non-essential amino acids. What are the essential amino acids and what are the non-essential amino acids? So the amino acids which cannot be synthesized in the body and must be supplied through diet are known as essential amino acids. Okay, again I repeat it. Essential amino acids means the amino acids which cannot be synthesized in the body and must be supplied through diet. Okay, these are called essential amino acids. Okay, so essential amino acids example: tryptophan, valine, lysine, histidine. So examples: tryptophan, valine, lysine, histidine. These are the examples of the essential amino acids. And second one is non-essential amino acids. Non-essential amino acids which are synthesized in the body, which are the amino acid which are synthesized in the body is called non-essential amino acids. So these need not be supplied through diet. Okay, so non-essential amino acids cannot be supplied through diet, but essential amino acids are synthesized, cannot be synthesized and supplied through diet. These are the differences between the essential amino acids and non-essential amino acids. Okay, so non-essential amino acids examples: cysteine, glycine, alanine, aspartic acid. These are the examples of the non-essential amino acids. Sometimes amino acids and their classification, this asking like a short answer. So first to write the what is the definition of amino acid and classification of amino acid and example. Okay, next, physical properties. So physical properties, the color and physical properties of glucose is colorless crystalline substances with high melting points. Colorless crystalline substances with high melting points. Okay, so these amino acids are soluble in water. These amino acids are soluble in water. Exist as dipolar ions. Exist as dipolar ions. Dipolar ions means both. They contain both positive, proton donor, proton acceptor. That means acids and bases. Okay, so these are the dipolars. So in this dipolar, the important 2 marks question is Zwitter ion. What is meant by Zwitter ion? In aqueous solution, the carbonyl group, that means COOH group of amino acid, loses a proton which is being accepted. The proton which accepted by amine group, that is NH2 group, by formation of a dipolar ion. So dipolar ion, how to form? One of the carboxylic acid loses their proton and accepted by the amine group present in the amino acid by the formation of dipolar. Such type of dipolar is known as Zwitter ion. Okay. Next, this is neutral but contains both positive and negative charges. See here, this one of this amino acid we are taking. In this amino acid, amine group, COOH group both are present, but here carboxylic group, COOH group, hydrogen is loses and they are attached to the amine group of the NH2 by the formation of positive and negative charges. So such type of structures are polars are known as dipolars. It is also known as Zwitter ion. Okay, next point. Amino acids are amphoteric. Amphoteric means both act acid and base because they can accept as well as proton donate. Okay, again repeated. Amino acids are amphoteric means they can accept as well as donate a proton. For example, Zwitter ion we are taking. In this Zwitter ion, positive negative charges are there. So in these Zwitter ion reacts with water, particularly in presence of acid. Hydrogen group is attached to the COOH group. H plus is added to the COOH group. So COO minus is attract the hydrogen to form first equation, that is NH3CHR COOH. Okay, next this Zwitter ion reacts with base OH group. So OH group, OH minus, by the liberation of water molecule from NH3 plus, we get NH2CHR COO- as the second compound. This is total about the Zwitter ion. Okay, so very important for 2 marks. Next, configurations. All the amino acids except glycine. All the amino acids except glycine are optically active. Optically active compounds means they are plain polarized light. Okay, all natural amino acids are L configuration. One of the L amino acid configuration we are taking here. Next, the linkage, that means amide bond, that means CONH bond, which units various amino acids is called peptide bond. Here the linkage CONH bond is called peptide bond. So here 2 moles of amino acids we are taking. In these 2 amino acids, one of the COOH group, another NH2 group, by the liberation of water molecule to form one of the peptide linkage, that is amide bond will be formed. Amide bond means CONH group is the amide bond. Okay. Next, polypeptides. Polypeptides are classified into so many types in that dipeptide, tripeptide, polypeptides. A dipeptide contains 2 amino acids. So peptides are classified into dipeptides, tripeptides, polypeptides. A dipeptide contains 2 amino acids linked to each other by peptide links. So okay, these are the peptides classification. So these are the total about the amino acids. So this is the total about the amino acids. So are you understanding? Okay, thank you.